So one change in the syllabus, we're going to save street plate for Wednesday because the extra long lecture today, I just want to make sure we get through all the biochemical tests by like middle of the lab period on Wednesday, and then we'll just talk about street plate and look at some videos on it. All right, so we're going to start off with biochemical tests. We've already performed 2 biochemical tests, right? We've done catalase and nitrate reduction. And so remember that the purpose of a biochemical test is to determine if a particular enzyme is present. And how do we know if an enzyme is present?
If the product is there.
Right, so we look for the end products, right? If we find the end products, then we assume the bacteria have that particular enzyme. And for catalase, the end product that we looked for was what? What was the end product for catalase that we could see? Nobody remembers? I know, it would be more memorable if you'd actually done it, right, the experiment, but remember we observed for oxygen bubbles? And then for nitrate reductase, we look for the end product nitrite. So the main point is we look for the end product of the enzymatic reaction. If we find it, then we assume the bacteria have that enzyme. Now, one of the documents on the Blackboard site is this a biochemical test chart. And it still has the old lab experiment numbers on it. But it's a good summary of all the biochemical tests that we're talking about today and on Wednesday. And it summarizes what the substrates are, the products, the enzymes, what a positive test looks like. So it's important to learn this test chart so that it can help you with the biochemical test.
Professor?
Yeah, Christiana?
Are you sharing your
Oh, I meant to. So I'll do that now.
Thank you.
There we go. So here, right, so the biochemical test chart. So it's on the Blackboard page, but it does summarize a lot of the important points. Although one of the other handouts we're going to see in a little bit does an excellent job with a lot of detail about all the biochemical tests. But this is a summary chart that I created that I've been using for at least 20 years. So the first 3 biochemical tests that we're going to talk about are starch, fat, and gelatin hydrolysis. So these are hydrolysis reactions. So water is going to be used to split a molecule. So starch, we know a lot about starch, don't we? What kind of molecule is starch?
Is it glucose?
Well, that's what it's made out of, right? So a long chain of glucose molecules. So it's a complex polysaccharide. And what enzyme hydrolyzes starch? What enzyme breaks down starch? If you took 150 at Rockville, you know this.
Amylase.
Amylase, right? Amylase. And technically, amylase hydrolyzes starch to produce maltose. What kind of sugar is maltose?
Disaccharide.
A disaccharide, right? A disaccharide. And what is maltose made out of? I believe Rachel already told us.
Malt sugar.
But what kind of sugars?
Glucose?
Glucose, right? It's made of two glucoses bonded to each other. And then the cells will split the maltose into just glucose. So the ultimate end product is glucose. Now, the problem with starch is that it's a big molecule. Think of it as a piece of spaghetti. Are those bacterial cells going to just, suck up that piece of spaghetti? It's way too big. So they have to break down the starch outside of the cell first. So amylase is secreted into the external environment. So because it's outside the cell, we call it an extracellular enzyme or an exoenzyme. And that way the starch can be broken down into smaller pieces, which can then be absorbed by the bacterial cell. So the bacteria must secrete the amylase into the environment to break down starch. And then it can absorb the smaller pieces. Now, do you remember from 150 how we tested for starch?
Is it by using iodine solution?
Yep, right. Remember we had the iodine and we get a really dark, almost black color. And so that indicated that starch was present. So it's the same kind of thing we're going to do with the starch test. Now, fat is another large molecule, and we're talking about a triglyceride. And so what are the two components that make a triglyceride? What two components make a triglyceride?
Is it glycerol and fatty acid?
Yep, glycerol and fatty acids. And so since triglycerides are a type of lipid, the enzyme is lipase, which will hydrolyze the triglycerol into glycerol and fatty acids. And so when you separate the fatty acids from the glycerol, they become acidic. And so we can easily test for acid in the lab using a pH indicator. And in this case, it's neutral red. And so we'll see that if the fat gets hydrolyzed, the neutral red turns a red color. So that's how we know that the fat was hydrolyzed. But again, the triglycerides are large molecules. They must be digested on the outside of the cell first. So lipase is also an extracellular enzyme or an exoenzyme. And so lastly, well, we've just seen the hydrolysis of carbohydrate and fat. So what's left to use for energy is protein. Our source of protein is going to be gelatin. So of course, proteins are made of long chains of amino acids. And when you hydrolyze a protein, you just get amino acids. So intact proteins, right, the intact polypeptides, they can form a gel. Amino acids by themselves cannot form a gel. So we're going to look for the liquefaction of the gelatin, right? So will the gelatin liquefy? Because that means that it has been hydrolyzed into amino acids, and amino acids do not form a gel. Now there is one little problem, and that's gelatin has a very low melting temperature. In fact, at 37 degrees C, which is the incubator temperature, gelatin melts and liquefies. But that's what we're looking for, is did the gelatin liquefy? But the incubator causes it to liquefy. So after we incubate the gelatin tubes, we have to refrigerate them, cool them down. If the gelatin is still intact, it will go back to being solid. But if the gelatin has truly been hydrolyzed, it will stay liquid in the refrigerator. So that's how we actually do the gelatin test. After incubation, we have to refrigerate and cool it down. If the gelatin goes back to being solid, no hydrolysis. If it's liquefied, then hydrolysis has occurred. And again, proteins are big molecules, right? The cell cannot absorb them. It must break them down first on the outside. And so the enzyme that hydrolyzes gelatin is a special protease, right? Because proteases in general break down proteins. And the protease specific for gelatin is gelatinase, which must be secreted into the environment to break down gelatin into smaller pieces that the bacteria can absorb. So amylase, lipase, and gelatinase are all extracellular enzymes because all those substrates are big. It must be broken down on the outside of the cell first, and then the smaller pieces can be absorbed. So let's look at what we would have done. So starch hydrolysis, we actually use just an ordinary TSA plate with a lot of starch in it. And then we inoculate the plate with bacteria, we incubate it, and the next class we actually cover the agar with iodine. And if the starch is still present, it turns really dark, almost black. But if the starch has been hydrolyzed, there's no black color. So in the organism on the left, let's see, the organism is growing nicely, right? It's just a TSA plate, so most bacteria will grow on that just fine. But do you see any hydrolysis in the organism on the left?
No.
No, because it's all black, right? We see the growth on top, but all the agar is still black, so there's been no hydrolysis of the starch. The organism on the right, we see a bright clear zone around the growth. And in fact, not only is there no starch there, right, to react with the iodine, but the agar plate turns clear enough to see through. And so in lab, when we do this, one of the things that I have the students do is put their plates on top of a piece of paper with writing. If you can see the writing through the agar, then you know the starch has been hydrolyzed. But we can see the clear zone around the growth. So no starch is in the agar around the growth. It's all been hydrolyzed. And because there is a zone, that means the enzyme has been secreted into the agar, and it's destroying the starch. So normally we test 3 different organisms. We use E. coli, Bacillus cereus or subtilis, either one, and then Streptococcus bovis. Now, E. coli would look like the organism on the left. No hydrolysis. And it's not really that surprising because E. coli is primarily found in the large intestine, and very little starch will make it to the large intestine. And that's because we humans produce amylase as well, right? Where do humans produce amylase?
Like, is it in their saliva?
All right, that's one place. So at the very beginning, when we're chewing our food, we start the digestion of starches in the oral cavity. But the problem, once we swallow and everything goes into the stomach acids denature the amylase. And so the breakdown of starch stops. So once the contents is moved into the small intestine, the pancreas will then secrete more amylase, right, to continue the breakdown of starch. So basically, there should be little to no starch left to reach E. coli. So E. coli is a carboholic. It loves carbohydrates, but it likes the simpler sugars. So no surprise for E. coli. When it comes to Bacillus, either Cereus or Subtilis, they are amylase positive. Not a large zone like what we're seeing here, but there is a small zone around the Bacillus growth. And so Bacillus also does like to eat starch. And in fact, it's very common for Bacillus cereus to be found on rice because rice is starch and Bacillus eats starch. The organism that we use that is really strongly positive for amylase is Streptococcus bovis, and it would look more like the organism on the right, with a pretty large zone where the hydrolysis has occurred. So, but we would just have inoculated our starch plates, incubated them. In the next class, we'd add iodine to the plates, look for a clear zone, which indicated the starch had been hydrolyzed. So any questions on starch? So with fat, we use neutral red fat agar. And so with the neutral red fat agar, it's basically just a nutrient agar plate, which contains neutral red, a pH indicator, and vegetable oil. So vegetable oil is our source of triglycerides. Neutral red is a pH indicator. If the bacteria use the enzyme lipase, they will separate the glycerol from the fatty acids, and the fatty acids become acidic. And we can see that. We get red fat droplets. And so you can see the organism on the right, Staph aureus, is positive for lipase because we have red fat droplets here. But E. coli is negative. E. coli doesn't like to eat fat either. Which again, not a big surprise because most fat gets absorbed before it reaches the colon. So there's not much fat for E. coli to eat, so it just prefers, carbohydrates. But Staph aureus is positive for lipase. And again, that's not really a big surprise because where do you find Staph aureus? Where does it commonly live?
On your skin?
On your skin. And does your skin secrete oils?
Yes, it does.
Yes, so no surprise that staph can eat oil, right? So there's on the skin, and your skin is oily, right? You secrete oils, so staph can eat it. But we look for the red fat droplets, and that tells us that the fat, the triglycerides, were hydrolyzed. Lastly, we have gelatin.
Question, Professor.
Yes?
The Staph aureus, is the one on the left or the right?
On the right. It's labeled Staph aureus. Right, so on the right is Staph aureus. E. coli is on the left. And then for gelatin, remember the enzyme is gelatinase. If the bacteria produce gelatinase, they will hydrolyze the gelatin so that when we stick it into the refrigerator, it stays liquid because the gelatin has been broken down into amino acids. If no hydrolysis has occurred, the gelatin will be a solid in the refrigerator. And so the tube on top is still solid, right? These are laying on their sides or being tilted at an angle. And so we have gelatin that's not hydrolyzed on top, and then gelatin that has been hydrolyzed on the bottom. So you see the liquid is at an angle because we're tilting the tubes. So the key thing to remember is after incubation, you must refrigerate the gelatin tubes and cool them down. If the liquid stays, then the gelatin has been hydrolyzed. If the gelatin goes back to being solid, no hydrolysis. Anybody like Jell-O? Gummy bears? Marshmallows? all made of
Yeah.
gelatin. So where does gelatin come from?
Fish.
Well, not most often. It depends. If you get kosher food, then it does, right? So basically, leftover parts of animals that we don't eat, like bones, cartilage, and their hooves, their toenails, right? Just cook it all up, add sugar and food coloring, and yum yum, right? But since most gelatin does often include or come from pigs, if you're kosher, then they do use fish bones in order to make the gelatin. So, but most marshmallows are just regular gelatin, unless you buy kosher marshmallows, which are made from fish bones. But they actually do make vegan marshmallows, so no animal products at all. They're a little bit denser than regular marshmallows, but they taste pretty good. So, but gelatin, if you're eating gelatin, you're probably eating some toenails in there, along with cartilage. So isn't that yummy? So any questions about starch, fat, or gelatin hydrolysis? Okay, now we're going to start the second handout. And so the original Word document is there, but it's huge. It's like 120 megabytes, but you can edit it so you can add stuff to it as you go. But I've included a much smaller PDF file if you need something smaller to work with. But remember, you cannot edit the PDF, but it is a lot smaller. So we're going to start talking about a variety of biochemical tests today, and we'll finish this on Wednesday. We'll look at things like carbohydrate fermentation, indole production, methyl red, Voges Proskauer test, citrate, hydrogen sulfide, urease tests. So most of these tests are actually used to identify enterics, but the gram-negative rods, especially the indole, methyl red, VP, citrate, hydrogen sulfide, and urease are primarily used for gram-negative rods. Although you can use them for all bacteria, but commonly we use them for the gram-negative rods to help us to identify them. So we're going to start by talking about carbohydrate fermentation first. So I'll just mention specifically the indole, methyl red, VP, and citrate tests are part of a group of tests called MVIC, which are used to identify gram-negative rods. In particular, we can use them to differentiate E. coli from Enterobacter aerogenes. So starting off with carbohydrate fermentation. So a general end product for fermentation is some type of acid, like pyruvic acid. So we can easily test for carbohydrate fermentation using a pH indicator. In this case, it's going to be phenol red. And so the phenol red broth is a nutrient broth that only contains one type of carbohydrate. And it's the type that we choose to put in there. And here at Rockville, we use glucose, lactose, and mannitol. So we already have some experience with mannitol. Remember the MSA plates, right? The mannitol salt agar plates. And how did we tell if the bacteria were fermenting mannitol? What did you see?
There would be yellow.
Yellow, right? So we're going to be looking for yellow broth. But when it comes to phenol red, If the pH goes above 7, if it's alkaline, then the broth turns pink. And it can be an intense fuchsia pink. So you can tell pH in the tube using a phenol red, but we're primarily looking for yellow broth, and that indicates the production of acid. Now the 3 carbohydrates that we use, glucose, lactose, and mannitol, we actually color code those tubes because then it makes it easier to recognize what it is very quickly. And we normally do require students to learn the color coding. And it's just what we use at Rockville just to keep things organized. But very typically, the glucose tubes have a blue cap, lactose has a red cap, mannitol has a silver cap. So you could easily recognize, oh, it's got a blue cap, it's got to be glucose, or red cap lactose. Silver, we use silver for other things, so you're also looking at the broth to see that it has a durum fermentation tube inside, because we're also looking for the production of carbon dioxide gas. Because some bacteria, when they ferment to make acid, they also make gas. And that's another characteristic we can use to identify different bacteria. So acid with or without gas. So you can have acid without gas, but you cannot have gas without acid, right? So that doesn't exist. Now what we're going to do is look at some tubes, and we're going to score them. A is acid, G is gas. A negative means there's no acid or no gas. But if there's a higher pH and you see a pink tube, we'll use a K just to indicate that it's alkaline, right? That it's basic. So here we have 5 carbohydrate fermentation tubes. And let me make this a bit bigger. So in our first tube, it's still the original red color. And of course, no gas bubble. So how would you score this? Tube number 1. How would you score it?
A.
Huh?
A.
Well, it's red. Tube number 1 is red. So what does that mean?
So G.
Do you see a gas bubble in here?
Oh no. So K.
I don't see anything. So we'd be a negative, right? No acid, no gas. What about tube 2? So what would you say for tube 2?
No gas also.
Right, it's not yellow and there's no gas. So remember, orange is not yellow. And so again, negative. Now, when you do carbohydrate fermentation tests, it is important to look at the tubes within 48 hours. And the reason why, if the tube sits too long, you could get a false positive. It could eventually turn kind of yellow, but if that happens after 48 hours, it's generally considered negative. Also, if the tube turns yellow really quickly, right, because the bacteria are really fermenting that sugar and making that acid, they will eventually use up all of the carbohydrate that's in that tube. So no more acid will be produced. And these are organic acids, right, like pyruvic acid, which the cell will then start to consume. It will start to use up the acids and the pH will go back up. And so the yellow color will fade. It'll start to get red again. So you always want to read your tubes within 48 hours. So orange is a negative result. If it's still orange in 48 hours, it's negative. What about tube 3?
Negative, negative.
It's negative, but it's pink. What does pink indicate? What's the
Acidic.
pH? What's the pH?
Acidic.
Pink? Pink is alkaline. It's basic, right? Yellow is acid. And so what about tube number 4? Christiana, turn your mic off. So what about tube number 4?
A minus, A negative.
Well, we know it's acid. And then what do we see inside here?
Would be AG.
Right, we see a gas bubble. Right, everybody see the gas bubble in here? So it's acid and gas. And then the last tube, tube 5.
Is it A minus?
Yep, acid but no gas. So that's how we would score the tubes. Now what we would do, we're actually in the lab using 4 different bacteria, E. coli, Micrococcus luteus, Proteus vulgaris, and Staph aureus. And so we have 4 different bacteria, 3 different carbohydrate tubes. So each group would be inoculating 12 tubes. So there's a lot of labeling involved. So you have to label all the tubes with what organism. And because you guys are not sure enough yet, you would label them with what carbohydrate was in there. But you would have 4 glucose tubes, 4 lactose tubes, and 4 mannitol tubes, right? Because we have 4 different bacteria. And we would have inoculated them using sterile swabs. And the reason for that is just to make the process go faster. Rather than using the loop and sterilizing over and over again to re-inoculate over and over again, we would use swabs, because that way you dip the swab in the bacterial culture and then just dip it into each carbohydrate tube. Just dip, 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 and you were done inoculating. So it was much faster doing it that way. And so that's how we would have done it. And then in the next class, you would have looked at the results and scored them for acid with or without gas or nothing at all, right? So like what we did with these sample tubes, you would have gone through to score your results. And you would have seen something very interesting. Like E. coli, I told you E. coli is a carboholic. Every tube would have been acid and gas for E. coli. Loves carbohydrates, very gassy, which is no surprise, right? So E. coli makes a lot of gas and loves carbohydrates. So every tube, acid and gas. Now, Staph aureus also does like a variety of carbohydrates, but does not produce any gas. So all the staph tubes would have been yellow, but no gas. Proteus vulgaris only ferments glucose, nothing else. So only the glucose tube would have acid in it, maybe a small gas bubble. And the gas bubbles do need to be significant. So sometimes there'll be just a little tiny gas bubble, so not really significant enough, but negative for everything else. And then the organism that was confusing was Micrococcus luteus. Because when you looked at all the tubes from Micrococcus, what you saw was negative, negative. Every tube was negative, all still red. And so Micrococcus did not ferment anything, not even glucose. How can that be? How can you have an organism that cannot even ferment glucose? And you actually do know the answer to this, but it's an application question. So Micrococcus luteus, all the tubes would have looked like tube number 1, all negative, no acid, no gas. Even for glucose, no acid, no gas. And so how can you have an organism that cannot ferment anything, not even glucose? Any ideas?
Is it like the lac operon?
Remember, glucose metabolism is constitutive, right?
Oh, right. I was thinking maybe they don't ferment glucose until something else is used up. But
No, even if glucose is the only carbohydrate in the tube, which it is, right? No other carbohydrates except glucose, Micrococcus will not ferment it. So how can you have an organism that doesn't even ferment glucose? So you know something about Micrococcus, right? Think back to the oxygen experiments. What did you learn about Micrococcus luteus?
they're obligate aerobes.
Right, they're obligate aerobes. That means they must have oxygen. And what do you know about fermentation?
The oxygen isn't the electron acceptor.
Right, so fermentation is an anaerobic process. So why would an obligate aerobe be able to perform an anaerobic process? It can't, right? So Micrococcus luteus cannot ferment anything because that's anaerobic. And Micrococcus cannot do anything anaerobic. So is that clear to everyone? It's a good application type question, right?
Professor, say it again, please.
So Micrococcus luteus is an obligate aerobe. It must have oxygen. We know that fermentation is an anaerobic process. So an obligate aerobe cannot perform an anaerobic process. Only oxygen is used. So no fermentation in Micrococcus luteus or any obligate aerobe. So does that make sense?
Yeah, thank you.
Okay, so that was carbohydrate fermentation. And of course, when you were finished with your swabs, you had to put them in the biohazard bags, right? So we could sterilize them. So what is the cause of a pink color to the phenol red broth as shown in tube 3? So remember, we have one tube here which is pink. So what do we say that means?
Is it because it was red after 48 hours?
Well, it went from red to pink. So what would cause that?
Because it's basic or
Right.
alkaline.
Yep. Alkaline pH. So number two, ultimately, what is the reason why different species of bacteria have different carbohydrate fermentation signatures? So why is it that everything cannot ferment everything?
Because of their oxygen requirements.
Right, that's one possible explanation, right? Oops. I'll correct that. Requirements. And also, when it comes to being able to use carbohydrates, what specifically do the bacteria need? And I think, Rachel, you already mentioned this. Like Proteus vulgaris, it can ferment glucose just fine, but not lactose, not mannitol, because it doesn't have what?
The operon.
Yep. Right, because there's operons for lactose, mannitol, maltose, sucrose, all kinds, right? So some bacteria have them, some don't. So for what purpose does a bacterial cell use carbohydrate fermentation?
Is it as a source of energy?
Yep. To make ATP. And again, using glycolysis. And so that helps to answer number 4. Does this relate to glycolysis? Of course, right? Fermentation uses glycolysis, right, to start the process. So if two species have identical carbohydrate fermentation signatures, would you still be able to differentiate them based on the production of different end products? So first, yes or no, can you differentiate two different fermenting bacteria based on their end products?
Yeah, by the bubbles, maybe?
Well, yes, because right, so if they produce different end products, you can test for them. So like alcohol fermentation versus lactic acid fermentation, right? So only if the end products are different. If they're the same end products, then there's really no way to tell. But if they do produce different end products, you can test for the end product. And so you can identify different organisms. And the example was lactic acid, versus alcohol fermentation, right? So easy to tell for lactic acid fermentation with pH. Alcohol fermentation, well, there are different things you could do to test for alcohol. But if they did happen to produce the exact same end product, then no, you could not differentiate them. But if the end products are different, then yes, you can differentiate them. So questions? Okay, we'll finish just talking about indole, and then we'll do the rest of the tests on Wednesday and the street plate. So indole is actually one of the 4 tests of the IMVIC test, so indole, methyl red, VP, and citrate. So indole is an end product. And originally scientists looked to see if bacteria made different kinds of end products as a way to help identify them. And so indole was an end product that was discovered that some bacteria make, and it's relatively easy to test for the presence of indole. So indole is made from tryptophan, and tryptophan is an amino acid. And so the enzyme that converts tryptophan into indole is tryptophanase. Imagine that, right? So some bacteria produce the enzyme tryptophanase, which can break down indole or break down a tryptophan into indole. And then we can easily test for indole using Kovacs reagent. And this is Kovacs reagent, which does contain hydrochloric acid, so you have to be careful with it. But we're looking for the production of indole. Now, tryptophan, you probably have heard of tryptophan before, right? It's especially popular during the last week of November. Why is tryptophan popular? I know you've heard of tryptophan. Where have you heard of it? Somebody tell me. I know you guys know, you've heard it. Last week of November, what happens in this country?
Black Friday.
Well, no, not the very end, but just in general towards the last week.
Thanksgiving?
Right? Thanksgiving. And what do most people like to eat?
Turkey.
Turkey, which has a lot of tryptophan in it. It's what I call the warm and fuzzy amino acid because it makes you feel all relaxed and sleepy. But I prefer to get my tryptophan from milk and cookies. So indole is something that we can easily test for using Kovacs reagent. As we'll see, it produces a red color. It's actually more of a hot pink fuchsia color. And the Kovacs actually does not mix with the liquid broth. It actually sits on top. So it's a very easy test to perform. And so what you have to do is inoculate your tryptone broth. So tryptone broth has a lot of tryptophan in it. So you inoculate, you incubate, in the next class, you add the Kovacs reagent. And so what always happens during the unknown, because students are all excited, is they inoculate, a test, and then they immediately add the chemicals to that tube. It's like, but they just inoculated it with bacteria, right? So you see the problem. You always have to incubate, give the bacteria time to grow and to make the end product. In the next class, you add whatever chemicals you need to see that reaction. So for almost everything except for like catalase, you have to inoculate, incubate, and the next class, you do the test. You add the chemicals or see a color change. So here we have 2 tryptone broth tubes, and they were inoculated and incubated, and then Kovacs reagent was added to the tubes. And you notice that it sits on top. And we can clearly see that kind of red fuchsia layer in tube 2. So that's a positive test for indole. In the tube on the left, it's just the color of the Kovacs reagent, which is kind of a yellowish color, kind of an olive green color, and it just sits on top. So it's very easy to see a positive indole test with the Kovacs. It either turns this, bright, red color, or it doesn't. So in tube 1, obviously it's negative. In tube 2, it's positive, right? Because we have that beautiful red, hot pink fuchsia layer on top. Now, we normally only test 2 bacteria, E. coli and Enterobacter aerogenes. And those are 2 very common gram-negative rods for the IMVIC test. And so it's easy to differentiate them based on these 4 different tests. But we're just going to stop here with the indole test for right now. Now, here's an interesting question. So the question is, since tryptophan is an essential amino acid, it's very important. It's a fairly complex molecule for an amino acid. Why would bacteria want to break it down and create indole? I mean, if you have something that's really important, like tryptophan, why do you want to waste the energy to make an enzyme and then tear that amino acid apart to make something like indole? Because as I said before, scientists just looked to see what kinds of things bacteria were making so that they could test for them, and indole was one of those products that they could easily test for. But the question is, why do they make indole at all? Why would they want to do that? And this has a typo in it. Possess. How do you spell possesses? Possesses. Oh, there we go. So, why would bacteria want to make indole from an essential amino acid? There must be a reason, right? So, at this link here is a good review paper, and I already have it up here. So, you should go and just sort of look through this. Well, the abstract kind of tells you sort of the key information, as does the title. Indole affects biofilm formation. Remember, biofilms are the result of the production of a lot of capsules and slime layers, and so that helps to protect those bacteria that are all living together in this biofilm. It's very protective. But indole does apparently have some important functions. So if you scan through here, I'll just sort of highlight key things here. So it is a metabolite of the amino acid tryptophan, and it's believed to function in things like virulence induction. So when we talk about disease and virulence, we'll learn that these are factors produced by pathogens to help overcome host resistance, the immune system, cell cycle regulation, acid resistance, and especially signaling biofilm formation. So one of the things that you'll do later is watch a video on quorum sensing. And quorum sensing is the ability of bacteria to talk to each other using chemical signals. And as you'll see, when lots of bacteria are all together, and they're all members of the same species, they're all secreting a chemical signal so they know that there's a lot of them all together, same time, same place. And so that allows them to turn on genes simultaneously. In that way, they can act like a large organism rather than a bunch of little individual organisms. And so apparently indole is a quorum sensing molecule and helps these bacteria to turn on genes to regulate the formation of biofilms. Right, so I'm just going to cheat. right? So, but make sure you take a look at that paper, just because that helps to highlight the idea that even though a long time ago scientists just found that some bacteria made indole and we can test for it, it turns out that the indole serves a purpose, right? Because why else would bacteria synthesize an enzyme to break down a complex essential amino acid? There's got to be a reason for it, right? It must serve a purpose. And so that paper helps to talk about, or helps to clarify the purposes by talking about them. So questions about indole? No questions? All right, then we're going to stop here for lab today. As I said, next time we'll finish the rest of the biochemical tests and also talk about streak plate and watch a video on streak plate. So,
Thank you.
all righty. Enjoy the cold, windy weather. At least the sun is shining, so that's good. All right, Clarice and Roxy, I'll see you guys on Wednesday.